Welcome to Bioinformatics.Expert. In the last video, we did a HiSat2 run to align RNA sequencing reads to a human genome. And in this video, I want to show you some programs which are necessary in the post-processing of the alignment file. The output of HiSat2, as well as the output of most of the other available alignment programs, is the so-called SAM format, which means sequence alignment file. However, the drawback of that file is that it is extremely large. For example, in our case, we see that it is 9 times larger than the raw data file. But we use only a small file with a reduced number of reads. If we would use a regular FASTQ file, which is much larger, then of course the output after the alignment would be extreme. Therefore, the first thing should be to reduce the file size, for example by converting the file type. The program of choice is samtools. This program not only converts the SAM format to the much smaller BAM format, which means binary alignment file, it also can sort the alignment results according to the genome coordinates. This makes it easier for the following programs and mostly is also a requirement for the following analysis steps. To start it, select it from the table. Now select the SAM file from the list. In the parameter table, select as tool parameter sort and as output BAM to convert it to the BAM format. Finally, start the analysis. After the analysis has finished, we see the BAM file. And when we go to the data repository, we see that the size of the file is reduced compared to the SAM format. That means that we can now delete the SAM file from the repository to free some disk space. With the sorted BAM file, we can now run the program picard mark duplicates. This program can be used to mark or to remove duplicated reads. Before we start, I want to emphasize that this program is actually an optional step. The scientific community is still arguing if it is necessary or not. In my opinion, it depends on the experiment and the output which we expect. However, as an example, we will test it here. Start the program and select the sorted BAM file from the file repository table. In the parameter table, we see that we have for the tool parameter 3 options: mark duplicated reads only, remove optical duplicates, and remove all duplicates. When we mark the duplicated reads only, then the program will just add an information to the BAM file. When we select remove all duplicates, then the program will create a new BAM file in which all duplicated reads are removed. However, when we have an experiment in which the output consists of peaks which are located in distinct genomic regions, then we cannot be sure if the duplicated read is a PCR artifact or part of the result. That means when we remove all duplicated reads, that high peaks might lose some of their intensity. That is why I don't like this option very much. However, you can of course decide by yourself and you can use each parameter as you wish. This option I like much more. Optical duplicates are reads which are actually in close proximity on the flow cell. That means when they are duplicated, then this is most likely due to technical artifacts of the sequencing procedure. So to remove them is quite a good idea. After the run has finished, there is a new BAM file. When we look at the output of the program, we see that actually no optical duplicates were found. This is of no surprise because we used a very small truncated raw data file which does not represent the complete flow cell of the experiment. With the final BAM file, we have now several options to continue. We can, for example, count the reads now with the program featureCounts. This is of importance when we want to do a differential gene expression analysis with the programs DSEQ2, DSEQ, or Etcher. We also can use the program stringT to detect new isoforms or calculate the TPM and FPKM values from the BAM file. However, I will show you stringT in a different video. So let's continue with featureCounts. First, select the final BAM file. In the parameter table, you can now select which feature you want to have in the final table file, and we can select the reference. In the backend of the program, the reference files are given as GTF files, which are necessary for featureCounts. You have to make sure that you use the same reference as you have used in the alignment step. featureCounts is now counting the reads which overlap specific features which are given in the reference file. We see that we actually get a quite small percentage of successfully assigned alignments. It's only 46.8%. However, you have to keep in mind that this percentage is calculated from all reads of the BAM file, not only from the aligned reads. Also, featureCounts does not count reads which overlap more than one feature. That means all reads which, for example, bridge to exons are discarded. Also, this is not very intuitive. It is very important for the differential gene expression programs like DSEQ2. At the end, featureCounts creates a list of raw counts which are saved as a text file, and this file can then be used in the differential gene expression analysis programs. But I will show you those in the next video. Alright, thank you for watching this video about the post-processing steps after an RNA sequencing alignment. Today we have seen the programs samtools, picard mark duplicates, and featureCounts. I also want to emphasize that I can add more programs upon request. If you want to use bioinformatic tools online in your research, then visit my webpage or write me an email.